Welcome to this peaceful journey through one of science's most profound mysteries, the origin of life on Earth. As you relax and let your mind wander through these fascinating concepts, allow yourself to drift into a state of calm contemplation. The story of how life began is a tale that spans billions of years, combining chemistry, physics, geology, and biology into one magnificent narrative. Let's begin our exploration by traveling back in time, back to when our planet was young, inhospitable, and yet somehow destined to become the cradle of life as we know it. Around 4.5 billion years ago, Earth was a very different place from the world we know today. The young planet was a hellish landscape of volcanic activity, meteor bombardments, and extreme temperatures. There were no oceans as we know them, no atmosphere suitable for breathing, and certainly no living organisms. Yet within this chaos, the building blocks of life were being assembled through natural chemical processes. The early Earth's atmosphere was likely composed of methane, ammonia, water vapor, and hydrogen, quite different from today's oxygen-rich air. Lightning strikes were frequent and powerful, providing immense amounts of energy that could drive chemical reactions. Ultraviolet radiation from the sun bathed the planet's surface without the protective ozone layer we have today. Volcanic eruptions constantly released gases and minerals into the environment. In this extreme setting, simple molecules began to interact in increasingly complex ways. Scientists believe that life's emergence required several key ingredients: liquid water, energy sources, and the right chemical building blocks. As Earth cooled over millions of years, water vapor in the atmosphere condensed to form the first oceans. These primordial seas became vast chemical laboratories where countless reactions occurred simultaneously. The water provided a medium for molecules to dissolve, move around, and encounter each other. It also offered protection from harsh ultraviolet radiation that would destroy delicate organic compounds on land. Energy was abundant in many forms, including lightning, ultraviolet light, heat from volcanic vents, and radioactive decay. This energy drove the formation of more complex molecules from simpler ones. One of the most famous experiments demonstrating how life's building blocks could form was conducted by Stanley Miller and Harold Urey in 1952. They created a sealed apparatus containing water, methane, ammonia, and hydrogen, then applied electrical sparks to simulate lightning. After just 1 week, they found that amino acids, the building blocks of proteins, had formed spontaneously. This groundbreaking experiment showed that organic molecules necessary for life could arise from simple inorganic precursors under early Earth conditions. The Miller-Urey experiment was revolutionary because it demonstrated that the gap between non-living chemistry and the chemistry of life was not as vast as previously thought. However, creating amino acids is just the first step. Life requires much more than amino acids. It needs nucleotides for genetic material, lipids for cell membranes, and complex molecules that can catalyze chemical reactions. The formation of these molecules and their organization into living systems remains one of science's greatest puzzles. Scientists have proposed several theories about where and how life originated. One popular hypothesis suggests that life began in shallow pools or tidal zones, where cycles of wetting and drying could concentrate organic molecules and promote their chemical bonding. Another theory points to deep-sea hydrothermal vents as the birthplace of life. These underwater volcanic springs release mineral-rich, heated water into the cold ocean. The chemical gradients and energy available at these vents create ideal conditions for complex chemistry. Modern hydrothermal vents support thriving ecosystems even in complete darkness, powered by chemical energy rather than sunlight. Some of Earth's most ancient microorganisms are found near these vents, suggesting a possible connection to life's origins. The hydrothermal vent hypothesis has gained considerable support because these environments provide several advantages. They offer stable conditions protected from surface bombardment by asteroids. They provide minerals that could catalyze reactions. And they create natural chambers in porous rocks where molecules could concentrate. The temperature gradients at vents could drive chemical processes similar to those in living cells. Additionally, the chemistry at these vents produces hydrogen and other compounds that could serve as energy sources for the first life forms. Another intriguing possibility is that life's raw materials came from space. Meteorites and comets contain organic compounds including amino acids, suggesting that the building blocks of life are common throughout the universe. Some scientists propose that life itself, or at least complex organic molecules, arrived on Earth from elsewhere in a process called panspermia. While this idea doesn't solve the ultimate question of how life originated, it expands the possible locations and conditions under which life could begin. Regardless of where the organic molecules came from, the next crucial step was their organization into self-replicating systems. This is perhaps the most mysterious part of life's origin. Modern life uses DNA to store genetic information, RNA to transfer that information, and proteins to do most of the chemical work. But this system presents a chicken-and-egg problem. DNA needs proteins to replicate, but proteins need DNA to be made. How could such interdependent molecules arise together? The RNA world hypothesis offers an elegant solution to this puzzle. RNA is a remarkable molecule that can both store genetic information like DNA and catalyze chemical reactions like proteins. Scientists have discovered that certain RNA molecules, called ribozymes, can actually copy other RNA molecules. This suggests that early life might have been based entirely on RNA, with DNA and proteins evolving later. In an RNA world, these versatile molecules would handle both genetics and metabolism. RNA molecules that were better at copying themselves would become more common, beginning the process of evolution by natural selection. Over time, this RNA-based life could have evolved to use DNA for more stable information storage and proteins for more efficient catalysis. Support for the RNA world hypothesis comes from several observations. Modern cells still use RNA in crucial roles including protein synthesis and gene regulation. Some viruses use RNA as their genetic material. The building blocks of RNA, called ribonucleotides, can form spontaneously under plausible early Earth conditions. Scientists have even created RNA molecules in the laboratory that can copy themselves without any protein enzymes. However, the RNA world hypothesis also faces challenges. Pure RNA is unstable and breaks down quickly in water. The spontaneous formation of long RNA molecules is difficult. Creating the right sequence of RNA building blocks by chance seems improbably rare. These difficulties have led researchers to consider whether even simpler genetic systems might have preceded RNA. Some scientists propose that life began with simpler molecules that later evolved into RNA. Peptide nucleic acids, or PNAs, are one possibility. These molecules have a protein-like backbone, but can store genetic information like nucleic acids. Threose nucleic acids, or TNAs, are another candidate. These alternatives might have been easier to form on early Earth and could have evolved into the RNA world we hypothesize existed. The transition from simple chemical systems to true life required the development of cell membranes. Modern cells are enclosed by lipid bilayer membranes that separate their internal chemistry from the external environment. This compartmentalization is essential for life because it allows different chemical concentrations to be maintained inside and outside the cell, enables the cell to control what enters and exits, and protects delicate molecules from the environment. Fortunately, the formation of simple membranes is relatively straightforward. Certain organic molecules called lipids spontaneously form hollow spheres called vesicles when placed in water. These protocells could have enclosed self-replicating RNA molecules, creating the first primitive life forms. Experiments have shown that protocells can grow, divide, and even compete with each other for resources. They can incorporate nutrients from their environment and expand as their internal chemistry produces more membrane material. When they become too large, they can split into two smaller protocells. This spontaneous behavior resembles cell division in modern organisms, though it's much simpler and doesn't require complex protein machinery. The earliest true life forms, once they emerged, would have been very simple compared to modern cells. They likely lacked most of the sophisticated molecular machinery we see in contemporary organisms. These primitive cells probably obtained energy and raw materials from their environment through simple chemical reactions. They may have fed on organic molecules that formed abiotically, meaning through non-biological processes. This lifestyle is called heterotrophic, depending on external sources of organic compounds. However, this lifestyle faced a fundamental problem. As these early organisms consumed the available organic molecules, they would have depleted their food source. This crisis may have driven one of evolution's most important innovations, photosynthesis. Some early cells developed the ability to harness sunlight, to create their own organic molecules from simple inorganic ingredients like carbon dioxide and water. This autotrophic lifestyle, making one's own food, freed these organisms from dependence on dwindling supplies of abiotic organic compounds. The evolution of photosynthesis had profound consequences for Earth. Early photosynthetic organisms released oxygen as a waste product. At first, this oxygen was absorbed by dissolved iron in the oceans, creating massive deposits of rust that we can still see today as banded iron formations. Once the ocean's iron was saturated, oxygen began accumulating in the atmosphere. This great oxidation event, which occurred around 2.5 billion years ago, fundamentally changed Earth's chemistry and opened up new possibilities for life. Oxygen is a highly reactive molecule that can damage cells, but it also enables much more efficient energy production. Organisms that evolved ways to use oxygen could extract far more energy from food molecules than those relying on fermentation or other anaerobic processes. This energy abundance may have enabled the evolution of more complex life forms. The first life on Earth was certainly prokaryotic, meaning cells without a nucleus or other internal compartments. Modern bacteria and archaea are prokaryotes, and their simplicity and versatility reflect the ancient origins of these domains of life. For roughly 2 billion years, prokaryotes were the only life on Earth. They diversified into countless forms, adapted to every conceivable environment, and fundamentally altered the planet's geology and atmosphere. Then, around 2 billion years ago, a remarkable event occurred: the evolution of eukaryotic cells. Eukaryotes have nuclei that house their DNA, along with other internal structures called organelles that perform specialized functions. This cellular complexity enabled the evolution of multicellular organisms including all plants, animals, and fungi. The origin of eukaryotic cells appears to have involved a process called endosymbiosis. In this scenario, one prokaryotic cell engulfed another, but instead of digesting it, the two cells formed a mutually beneficial relationship. The engulfed cell became an organelle, providing services to its host. Mitochondria, the power plants of eukaryotic cells, were likely once free-living bacteria that took up residence inside another cell. They retained their own DNA and can still reproduce independently within cells. Chloroplasts, the photosynthetic organelles in plant cells, have a similar origin from ancient cyanobacteria. Evidence for endosymbiosis is compelling. Mitochondria and chloroplasts have their own circular DNA, similar to bacteria. They have double membranes, consistent with being engulfed by another cell. They reproduce by dividing, just like bacteria. Their ribosomes, the molecular machines that make proteins, resemble bacterial ribosomes more than eukaryotic ones. This evidence paints a picture of eukaryotic cells as communities of formerly independent organisms that merged into a single entity. The earliest evidence of life on Earth comes from several sources, each with its own strengths and limitations. Fossilized microbial mats called stromatolites date back 3.5 billion years. These layered structures are formed by communities of microorganisms that trap sediment and build up over time. Modern stromatolites still form in places like Shark Bay in Australia, allowing scientists to study how ancient ones might have developed. Chemical signatures in ancient rocks provide another line of evidence. Carbon isotopes in rocks as old as 3.8 billion years show patterns consistent with biological activity. Living organisms preferentially use lighter carbon isotopes, leaving a distinctive signature in organic matter. Finding this signature in ancient rocks suggests that life was present when they formed. However, interpreting these chemical fossils requires caution, because non-biological processes can sometimes produce similar patterns. The most direct evidence of early life comes from microfossils, preserved remains of ancient microorganisms. Scientists have found structures that appear to be fossilized bacteria in rocks from Western Australia, dated to 3.4 billion years ago. These microscopic fossils show cell-like shapes and sizes consistent with prokaryotes. Some even occur in filaments similar to modern bacteria. However, not all scientists agree that these structures are biological. Distinguishing ancient microfossils from mineral formations that coincidentally resemble cells is challenging and controversial. Despite these challenges, the consensus among scientists is that life appeared on Earth relatively quickly after conditions became suitable. The planet formed 4.5 billion years ago. The Late Heavy Bombardment, a period of intense asteroid impacts, ended around 3.8 billion years ago. Evidence of life appears in rocks just a few hundred million years later. This rapid emergence suggests that life might form readily when the right conditions are present. The question of how common life is in the universe depends partly on understanding how it originated on Earth. If life requires an extremely unlikely series of coincidences, we might be alone or nearly alone in the cosmos. If life arises naturally wherever conditions permit, the universe could be teeming with living worlds. Scientists are searching for life elsewhere in our solar system, with Mars and some moons of Jupiter and Saturn being prime candidates. Finding even simple microbial life on another world would revolutionize our understanding of life's origin and prevalence. The process by which non-living matter becomes living is called abiogenesis. While we've discussed many hypotheses about how this might have occurred, we still don't have a complete, step-by-step explanation. This gap in our knowledge is not surprising given the enormous time scales involved and the difficulty of recreating early Earth conditions in the laboratory. However, research continues to make progress. Scientists are working to understand how each component of living cells could have arisen through natural processes. They're investigating how genetic information could emerge from chemistry, how metabolism could begin without enzymes, how cells could form and reproduce, and how all these systems could integrate. One fascinating area of research focuses on chemical networks that can maintain themselves. Modern cells have elaborate metabolic pathways where hundreds of chemical reactions are carefully coordinated. Each reaction is catalyzed by a specific enzyme protein. But how could such complexity arise gradually? Researchers have found that some mineral surfaces can catalyze organic reactions without proteins. Iron and sulfur minerals, common at hydrothermal vents, can promote the formation of organic molecules. These mineral catalysts might have served as protoenzymes, speeding up reactions in prebiotic chemical systems. As these systems evolved, RNA or protein catalysts could have gradually replaced mineral ones, leading to the biochemistry we see today. Another important consideration is energy flow. All living things require energy to maintain their organization and grow. Modern cells use sophisticated molecular machines to capture and utilize energy. The earliest life forms must have tapped into energy sources available on early Earth. Chemical gradients at hydrothermal vents, where hot acidic water meets cooler alkaline ocean water, could have provided such energy. The flow of protons across natural mineral membranes could have driven the first metabolic reactions. Some scientists propose that life emerged within the microscopic pores of these mineral structures before developing its own membranes. The definition of life itself becomes blurry when considering its origins. What distinguishes a complex chemical system from a true living organism? Most definitions of life include certain characteristics: organization into cells, metabolism to acquire and use energy, growth and reproduction, response to environment, and evolution through natural selection. But these criteria didn't all appear simultaneously. Early life probably had some of these properties but not others. There must have been transitional entities, neither clearly living nor clearly non-living. Scientists sometimes call these hypothetical systems protocells or protolife. Understanding this gray zone between chemistry and biology is crucial for understanding life's origin. The role of chance versus necessity in life's origin is another deep question. Some aspects of life may be chemically inevitable under the right conditions. The formation of membranes from lipids, for instance, follows from the basic physics of how these molecules interact with water. Other aspects, particularly the specific sequences of genetic information, likely involved substantial randomness. The first self-replicating molecule probably had a particular sequence that worked, but many other sequences might have worked equally well. Evolution through natural selection then shaped and refined these early systems. As we relax and contemplate these vast timescales and profound questions, it's worth reflecting on the connections between all living things. Every organism on Earth, from bacteria to blue whales, shares a common ancestor. We all use the same genetic code with only minor variations. We all use ATP as an energy currency. We all build our proteins from the same set of 20 amino acids. These universal features of life point to a single origin event, one population of early cells that gave rise to all subsequent life. This unity of life is one of biology's most profound insights. The tree of life branches from this common ancestor into three great domains: bacteria, archaea, and eukarya. Bacteria and archaea are both prokaryotes, but differ in their biochemistry and genetics. Archaea are particularly notable for thriving in extreme environments like hot springs, salt lakes, and acidic waters. These extreme conditions might resemble environments on early Earth, making archaea particularly interesting for understanding life's origins. Studying modern microorganisms that live in extreme environments, called extremophiles, gives us insights into the possible conditions under which life began. Some bacteria thrive at temperatures above the boiling point of water near hydrothermal vents. Others survive in the frozen lakes of Antarctica or deep underground in solid rock. These discoveries have expanded our conception of where life can exist, both on Earth and potentially on other worlds. The search for life's origin is not just a scientific question, but also a philosophical one. Understanding how life began addresses fundamental questions about our place in the universe. Are we the result of cosmic purpose or fortunate chance? Is life rare and precious or common and abundant? These questions touch on meaning, value, and our relationship with the cosmos. Science cannot answer all philosophical questions, but it can inform them with evidence and constrain the range of possibilities. Modern origin of life research involves many scientific disciplines working together. Chemists synthesize organic molecules and study their reactions. Biologists examine the simplest modern organisms for clues about ancient life. Geologists analyze ancient rocks for chemical and fossil evidence. Astronomers search for planets that might host life. Physicists model energy flows in prebiotic systems. Computer scientists simulate the evolution of chemical networks. This interdisciplinary effort reflects the complexity of the question and the many types of evidence relevant to answering it. Experimental approaches continue to evolve. Beyond classic experiments like Miller-Urey, scientists now create artificial cells in the laboratory, trying to produce self-replicating chemical systems from scratch. They expose simple molecules to conditions, simulating various early Earth environments, and observe what compounds form. They study how information can arise in chemical systems without pre-existing genetic material. Each experiment adds another piece to the puzzle, gradually narrowing the gap between non-life and life. One particularly exciting area involves directed evolution experiments. Scientists can now design RNA molecules that perform specific functions, then let them evolve in the laboratory over many generations. These experiments show how molecular systems can increase in complexity and capability through variation and selection. While these experiments use intelligent design to set up initial conditions, they demonstrate that once self-replicating molecules exist, evolution can craft remarkable complexity without further intervention. The question of whether life on Earth originated here or arrived from space remains open. Panspermia, the idea that life or its precursors came from elsewhere, is testable to some degree. If we found life on Mars or Europa, and it used the same genetic code as Earth life, that would suggest a common origin, possibly through exchange of material between worlds. If we found life that used completely different biochemistry, that would suggest independent origins and imply that life arises readily when conditions permit. The discovery of thousands of exoplanets, planets orbiting other stars, has revolutionized our perspective on this question. We now know that planets are extremely common in the universe. Many orbit in the habitable zone where liquid water could exist. Some of these worlds might have conditions similar to early Earth. If life originated relatively easily on Earth, it may have done so on countless other worlds as well. Future telescopes may be able to analyze the atmospheres of distant planets, looking for chemical signatures that could indicate biological activity. As we drift peacefully through these concepts, consider the remarkable journey from simple atoms to living cells. Hydrogen, carbon, oxygen, nitrogen, and other elements forged in the cores of ancient stars came together on a young planet. Through natural chemical processes driven by energy from lightning, sunlight, and geothermal heat, these atoms organized into molecules. Those molecules formed larger structures. Some structures could copy themselves, Primitive cells enclosed these self-replicators. Evolution began, shaping and refining life over billions of years. The result is the magnificent diversity of life we see today, from microscopic bacteria to towering trees, from jellyfish to humans. We are made of stardust, organized through natural processes into thinking, feeling beings capable of pondering our own origins. The study of life's origin reminds us of the deep connections between chemistry and biology, between Earth's history and life's history, between our planet and the cosmos. It shows us that life emerges from and depends upon the physical and chemical properties of the universe. Understanding these connections enriches our appreciation of the natural world and our place within it. As research continues, we can expect our understanding of life's origin to become more detailed and complete. New discoveries in chemistry, biology, geology, and astronomy will fill in gaps in our knowledge. We may even achieve the remarkable goal of creating synthetic life in the laboratory, demonstrating conclusively that life can arise from non-living matter through understood processes. Such an achievement would not diminish the wonder of life, but rather deepen it. The origin of life represents one of the most dramatic phase transitions in the history of the universe. Matter organized itself in new ways, creating systems that could maintain themselves, reproduce, and evolve. This transition enabled the subsequent evolution of all the complexity we see in the living world. Understanding this transition is key to understanding life itself. The chemical processes that gave rise to life continue to operate in living cells today. The metabolism that keeps us alive is related to the chemistry that first energized primitive cells billions of years ago. Our genetic material, DNA, is a molecular descendant of the first self-replicating molecules. The membranes that surround our cells form through the same self-organizing properties that created the first protocells. Life is not separate from chemistry and physics, but is rather chemistry and physics organized in particularly interesting and complex ways. This perspective, far from making life seem less special, reveals its true magnificence. The same atoms that sit inert in rocks can, when properly organized, think and feel and wonder about their own existence. The emergence of life shows us that the universe has creative potential, that simple rules can give rise to extraordinary complexity, that matter can organize itself into patterns of stunning sophistication. These insights resonate with both scientific and spiritual dimensions of human experience. Throughout history, humans have wondered about the origin of life and our place in the cosmos. Ancient myths and religions offered various accounts of creation. Modern science provides a different kind of answer, one based on evidence and natural processes. These approaches need not conflict. Science tells us how life originated through material processes, while questions of meaning and purpose may extend beyond science's domain. Many people find that understanding the natural history of life enhances rather than diminishes their sense of wonder and appreciation for existence. As you continue to relax and let these ideas wash over you, think about the unbroken chain of life connecting you to the first cells that emerged in ancient oceans. Every human alive today is descended from those pioneering microbes. Our DNA carries echoes of that ancient heritage. The biochemical processes occurring in your cells right now have been refined over billions of years of evolution, but their fundamental logic was established by early life. You are part of a story that spans the age of Earth, a story of matter becoming alive and evolving into the rich diversity we see today. The search for life's origin continues to captivate scientists and laypeople alike because it speaks to fundamental questions about existence. How did something come from nothing? Well, life didn't come from nothing. It arose from pre-existing matter and energy through natural processes. But how did non-living matter become living? This question drives decades of research and thousands of experiments. Each small advance in our understanding is a triumph of human curiosity and ingenuity. Each answered question reveals new mysteries to explore. This ongoing process of discovery is one of humanity's noblest endeavors. Looking to the future, we can anticipate breakthroughs in origin of life research. Advances in technology allow us to probe chemical systems with unprecedented precision. Powerful computers can simulate complex chemical networks. New analytical techniques can detect minute quantities of organic molecules in ancient rocks. Space missions may bring back samples from Mars or icy moons, where prebiotic chemistry might be occurring. Earth-based experiments continue to recreate conditions of early Earth and observe what emerges. All these efforts contribute to a growing understanding of how life began. One intriguing possibility is that life may arise through multiple pathways. Perhaps there are several different routes from chemistry to biology. Early Earth may have seen multiple independent origins of life, with different chemical systems trying different solutions to the challenges of self-replication and metabolism. If so, modern life might descend from the most successful of these early experiments, while other paths died out without leaving descendants. Alternatively, all modern life might result from a single successful origin, with other attempts never quite achieving sustainable replication. We may never know with certainty which scenario occurred, but both possibilities are fascinating to contemplate. The concept of a last universal common ancestor, often abbreviated LUCA, is important in origin of life studies. LUCA was not the first living thing, but rather the most recent organism from which all current life descends. By comparing all modern organisms and identifying their shared features, scientists can infer properties of LUCA. This exercise tells us about the characteristics that ancient life possessed before the three domains diverged. LUCA apparently had DNA, RNA, and proteins, suggesting these features are very ancient. It likely lived in a hot environment, possibly near hydrothermal vents. It had a fairly sophisticated metabolism and could produce many of its own organic molecules. Between the true origin of life and LUCA, there must have been substantial evolution and increasing complexity. The field of astrobiology combines origin of life research with the search for life beyond Earth. By understanding how life began on our planet, we can better predict where else it might exist. Mars likely had liquid water on its surface billions of years ago, when life was starting on Earth. Did life also begin on Mars? If so, does it persist today in underground aquifers? Europa and Enceladus, moons of Jupiter and Saturn, have subsurface oceans that might harbor life. Titan, another moon of Saturn, has liquid methane lakes where exotic forms of chemistry occur. Each of these worlds represents a natural experiment in whether life can arise under different conditions. Exploring them will test our theories about life's origins and requirements. As you rest peacefully, let your mind wander through deep time, imagining the ancient Earth and the chemical reactions that eventually led to life. Picture the early oceans, filled with dissolved minerals and organic molecules, energized by lightning and volcanic heat. Imagine shorelines where waves splash over rocks encrusted with organic films. Visualize hydrothermal vents, columns of superheated water rising from cracks in the ocean floor, surrounded by gradients of temperature and chemistry. In one of these settings, or perhaps in several, chemistry became organized enough to sustain itself and reproduce. That moment, whenever and wherever it occurred, was the birth of life on Earth. From that humble beginning, evolution crafted everything from sequoia trees to butterflies, from coral reefs to human civilizations. The study of life's origin connects to many other areas of science and philosophy. It relates to questions about the nature of information and complexity. How does information arise in physical systems? How do complex patterns emerge from simple rules? These questions extend beyond biology to physics, computer science, and mathematics. They touch on ideas about self-organization, emergence, and the arrow of time. Life is perhaps the most remarkable example of emergent complexity in the universe, where the whole is far more than the sum of its parts. Understanding how this emergence occurred enriches our grasp of fundamental principles governing the cosmos. In closing this exploration of life's origin, we return to the sense of wonder that drives all scientific inquiry. 4.5 billion years ago, our planet formed from a disk of dust and gas surrounding the young sun. After cooling and the formation of oceans, chemistry began creating increasingly complex molecules. At some point, a system arose that could make copies of itself. This self-replication, combined with variation and selection, initiated evolution. Over billions of years, evolution generated the magnificent pageant of life we see today. We are part of that pageant, products of the same natural processes that created the first cells. Understanding our origins through science doesn't diminish the wonder of existence, but rather deepens it. We are the universe becoming conscious of itself, matter organized in such a way as to contemplate its own history and origin. As you drift toward sleep, let these ideas settle gently in your mind. The origin of life is a story of time, chemistry, energy, and chance. It's a story that continues to unfold as scientists make new discoveries and refine theories. It's a story that connects us to the cosmos and to every living thing on Earth. It reminds us that we are part of nature, not separate from it, and that the same laws governing stars and atoms also govern cells and ecosystems. This unity of nature, this connection between all things through natural processes, is one of science's most profound revelations. Rest well, knowing that you are part of an ancient lineage stretching back to the dawn of life itself, and that the atoms in your body have been on an incredible journey through space and time to become you. Let us continue exploring this fascinating topic in greater depth, examining the molecular machinery of life and how it might have originated. The genetic code, used by all known life, is a mapping between sequences of 3 nucleotides, called codons, and the 20 amino acids used to build proteins. This code is nearly universal, with only minor variations in a few organisms. The universality of the genetic code strongly suggests that all life descended from a single ancestor that used this particular code. But how did this code originate? Why these particular codons for these particular amino acids? Scientists have discovered patterns in the genetic code that suggest it evolved to minimize errors. Mutations often change one nucleotide in a codon. The genetic code is organized so that such mutations frequently result in chemically similar amino acids being substituted, reducing the harmful effects of mutations. This error minimization suggests that the code was refined by natural selection over long periods. Early life forms with less optimal codes would have suffered more from mutations and been outcompeted. The specific assignment of codons to amino acids may have started with a simpler code using fewer amino acids. As evolution proceeded, new amino acids were incorporated and new codons assigned. Some patterns in the modern code hint at this history. For instance, amino acids with similar chemical properties often have related codons, suggesting they were added to the code at similar times. The evolution of protein synthesis is another fascinating puzzle. Modern cells use elaborate molecular machines called ribosomes to build proteins according to genetic instructions. Ribosomes are huge complexes of RNA and protein that coordinate the assembly of amino acids in the correct order. Transfer RNAs carry amino acids to the ribosome and match them to the appropriate codons in messenger RNA. This whole system is exquisitely precise and complex. How could it have evolved? The RNA world hypothesis suggests that early protein synthesis was carried out entirely by RNA molecules, with proteins added later to improve efficiency. The ribosome, even in modern cells, has an RNA core that performs the actual catalysis of peptide bond formation. The proteins in modern ribosomes play mostly structural and regulatory roles. This suggests that ribosomes are ancient RNA machines that later acquired protein components. Discovery of ribozymes, RNA molecules that catalyze chemical reactions, supports this view. Some ribozymes can even catalyze the formation of peptide bonds, the chemical links between amino acids in proteins. This shows that RNA alone could potentially have handled early protein synthesis, crude though it may have been. Metabolism, the network of chemical reactions that extract energy from the environment and build cellular components, must also have originated early. Modern metabolic pathways involve hundreds of enzymes working in coordinated sequences. Each enzyme is precisely shaped to catalyze a specific reaction. How could such intricate systems originate? One approach to this question focuses on core metabolic pathways that are universal among living organisms. The citric acid cycle, also called the Krebs cycle, is a central metabolic pathway used by virtually all organisms to extract energy from organic molecules. Some scientists propose that this cycle or something similar could have operated before life, driven by mineral catalysts at hydrothermal vents. If true, early cells might have inherited their core metabolism from prebiotic chemistry, rather than inventing it from scratch. This idea is called metabolism first, in contrast to the gene-first approach of the RNA world hypothesis. Metabolism first theories propose that life began with self-sustaining networks of chemical reactions that could maintain themselves by capturing energy and matter from their environment. Genetic information storage evolved later to make these systems more reliable and evolvable. Both perspectives have merit, and they're not mutually exclusive. Life likely required both information molecules and metabolic cycles from early on. Understanding how these systems integrated is a key goal of origin of life research. The physical chemistry of water plays a crucial role in life as we know it. Water has unusual properties that make it an excellent medium for biology. It's liquid over a wide temperature range under Earth's surface conditions. It's an excellent solvent for many types of molecules. It has high heat capacity, making temperatures more stable. Its density maximum at 4 degrees Celsius causes ice to float, insulating water below. All these properties make water uniquely suitable for life, at least life as it evolved on Earth. The hydrophobic effect, where oily molecules cluster together to minimize contact with water, is fundamental to biological structure. Cell membranes form because lipids have one water-loving end and one water-avoiding end. When these molecules encounter water, they spontaneously organize into double layers with the water-avoiding tails pointing inward and the water-loving heads pointing outward. This creates a barrier that can separate the cell's interior from the external environment. No complex molecular machinery is required. The structure emerges naturally from the physical properties of lipids and water. This self-assembly is likely how the first cell membranes formed, providing compartments where prebiotic chemistry could occur in a controlled environment. Inside these compartments, molecules could be concentrated, increasing reaction rates. Useful molecules could be retained while waste products might diffuse out or be actively expelled. The membrane provides a boundary defining what's self and what's environment, a fundamental property of living things. Modern membranes are not simple lipid bilayers, but complex structures containing many embedded proteins that control transport, harvest energy, and process information. But the first membranes were probably just simple lipid vesicles that happened to contain self-replicating molecules. Evolution then gradually elaborated this basic structure, adding complexity as it provided advantages. Chirality, or handedness, is another intriguing aspect of life's chemistry. Many molecules can exist in two mirror-image forms, like left and right hands. These are called enantiomers. Remarkably, life uses only one form of each type of molecule. Amino acids in proteins are all left-handed. Sugars in nucleic acids are all right-handed. This homochirality is essential for biological function. Proteins folded from mixed chirality amino acids wouldn't have consistent three-dimensional shapes. The origin of biological homochirality is not fully understood. In non-biological synthesis, left and right-handed molecules form in equal amounts. Some process must have selected one form over the other in prebiotic chemistry. Several mechanisms have been proposed. Circularly polarized light from stars or reflected from crystals might preferentially destroy one enantiomer. Certain mineral surfaces might preferentially bind one form. Once a slight excess of one enantiomer exists, autocatalytic reactions could amplify this asymmetry. Some molecules can catalyze their own formation and simultaneously inhibit formation of their mirror image. In experiments, researchers have seen small initial asymmetries grow into complete homochirality through such processes. Perhaps early Earth happened to develop a slight excess of left-handed amino acids through one of these mechanisms. Chemical evolution then amplified this small bias into the complete homochirality we see in modern life. If life originated multiple times independently, these origins might have chosen different chirality conventions. But once one convention was established in the lineage that led to all modern life, it became frozen in. Changing chirality would require rebuilding all cellular machinery simultaneously, an impossibly unlikely event. So we're stuck with the chirality choices made billions of years ago by the first evolving molecular systems. The concept of chemical evolution is important for understanding life's origin. Before there were living organisms that could undergo biological evolution, there were molecular systems undergoing chemical evolution. Molecules that were better at copying themselves became more abundant. Molecules that could catalyze useful reactions persisted while others broke down. These chemical selection pressures shaped early molecular systems, setting the stage for true biological evolution once cells emerged. Chemical evolution doesn't require cells or even well-defined individuals. It can operate on populations of molecules in solution or on surfaces. The key requirement is that some molecular variants increase in number faster than others, creating differential replication. Once you have differential replication, you have evolution. Natural selection will favor variants that replicate most efficiently under local conditions. This process can generate complexity even in the absence of biology. Computer simulations of chemical evolution show that complex autocatalytic networks can emerge from simple starting conditions. These networks can exhibit properties like adaptation and competition. They can evolve to become more efficient at capturing resources and replicating. While these simulations are simplified compared to real chemistry, they demonstrate that natural selection can operate at a molecular level, shaping systems toward greater complexity and efficiency. The timeline of life's origin remains uncertain. The oldest widely accepted evidence of life dates to about 3.5 billion years ago. Earth's surface became cool enough for liquid water perhaps 4 billion years ago. This leaves a window of several hundred million years for life to originate. Geologically, this is not a long time. It suggests that life arose relatively quickly once conditions permitted. However, some scientists argue for an even earlier origin. Carbon isotope evidence in rocks 3.8 billion years old might indicate biological activity. If confirmed, this would push back the origin of life to very soon after the late heavy bombardment ended. Some researchers even speculate that life might have originated multiple times, with early attempts wiped out by asteroid impacts before finally taking permanent hold. The pace of early evolution is also debated. Did life arise suddenly in a geologically brief period, or did the transition from chemistry to biology take millions of years? We don't know. The first self-replicating molecules might have appeared relatively quickly, but the evolution from simple replicators to cells with membranes, metabolism, and genetic material may have taken considerable time. This period of early evolution, before the last universal common ancestor, might have been when many biochemical features were established. Different lineages might have experimented with different solutions to biological problems. The lineage that ultimately succeeded may have had advantages in efficiency, robustness, or adaptability that allowed it to outcompete or absorb other early life forms. Horizontal gene transfer, where organisms acquire genes from other organisms rather than from ancestors, was probably very common in early life. Modern prokaryotes frequently exchange genetic material with distantly related organisms. This ability to share innovations may have accelerated early evolution, allowing beneficial adaptations to spread quickly through populations. It also complicates efforts to trace early evolutionary history, since genes have shuffled between lineages. The tree of life's deepest branches may be more like a web than a simple branching tree. Competition and cooperation both played roles in early evolution. Cells competed for resources, with more efficient variants outcompeting others, but cooperation also emerged. Cells that could work together or specialize in different functions could thrive in ways that isolated cells could not. This balance between competition and cooperation continues to shape evolution today. Life exists at multiple scales of organization, from individual molecules to whole ecosystems, and selection operates at all these levels. The role of contingency versus determinism in evolution is philosophically interesting. To what extent was life's specific path historically contingent on chance events? And to what extent was it determined by physical and chemical necessity? If we could rewind Earth's history and let it play out again, would life emerge? Would it look similar to what we have now? These questions are difficult to answer definitively. But studying life's origin helps us understand which features were likely necessary given physics and chemistry, and which were accidents of history. The universality of certain features, like the genetic code and basic metabolic pathways, suggests some degree of determinism. The enormous diversity in details suggests contingency also played a major role. A different sequence of random events might have led to quite different specifics, while preserving certain broad features. Synthetic biology, the creation of new biological systems and organisms in the laboratory, is closely related to origin of life research. By attempting to build living systems from scratch, scientists learn which components are essential and which are optional. They discover what minimal set of genes and functions a cell needs to survive. They test whether alternative biochemistries could support life. These experiments probe the boundaries of what counts as living and help us understand what was necessary for life's origin. Several research groups are working toward creating synthetic cells. These efforts involve designing minimal genomes that encode only essential functions, creating artificial cells enclosed in synthetic membranes, and combining genetic and metabolic systems into integrated wholes. While no one has yet created a fully synthetic cell that can grow and divide independently, progress is steady. Each advance teaches us about the requirements for life and the challenges that early evolution had to overcome. Some researchers are exploring whether alternative biochemistries could support life. Could life use silicon instead of carbon as its structural element? Could it use solvents other than water? Could it use different molecules to store genetic information? While speculative, these questions help us understand what's universal about life and what's specific to Earth's particular solution. They also help us think about what extraterrestrial life might look like if it evolved under different conditions. Carbon is favored for life because it can form 4 chemical bonds, allowing complex branching molecules. Silicon can also form 4 bonds, but silicon-based molecules are generally less stable and less diverse than carbon-based ones. Water is favorable because it's liquid over a useful temperature range and dissolves many substances. Alternative solvents like ammonia or methane have different properties that might support alternative biochemistries in very cold environments. These speculations aren't just academic. Titan, Saturn's largest moon, has lakes of liquid methane and ethane. If life exists there, it must use radically different chemistry than Earth life. Understanding what alternative biochemistries are possible helps us recognize life if we encounter it in exotic environments. The energetics of early life are crucial to understanding its origin. All living things require energy to maintain their organization against entropy's tendency toward disorder. They capture energy from their environment in various forms: sunlight, chemical gradients, or energy-rich molecules. The first life forms had to tap into whatever energy sources were available on early Earth. Likely candidates include chemical disequilibrium at hydrothermal vents, ultraviolet light, lightning, radioactive decay, and geothermal heat. Any of these could have driven the first metabolic reactions. Modern cells store energy primarily in the molecule ATP, adenosine triphosphate. This molecule has high-energy phosphate bonds that release energy when broken. ATP is used to power virtually all energy-requiring processes in cells. Interestingly, ATP is also a building block of RNA. This connection hints at ancient links between energy metabolism and genetic information. Perhaps the earliest life forms used RNA-related molecules both for information storage and energy currency. As evolution proceeded, specialized molecules took over different functions, but the connection remains visible in modern biochemistry. The amount of energy available in different environments affects what forms of life can exist there. High-energy environments like hydrothermal vents can support diverse metabolisms. Low-energy environments constrain what's possible. Understanding these energy constraints helps us predict where life might originate and what forms it might take. Some of the most energy-poor ecosystems on Earth are deep subsurface environments, where microbes live kilometers below the surface in rock pores. Growing extremely slowly on meager energy, supplies from radioactive decay and chemical reactions. These organisms show how little energy is necessary to maintain life. They may resemble early life forms that eked out existence on limited energy sources. The evolution of energy harvesting was a major driver of biological innovation. Early organisms that fermented organic molecules were limited by their food supply. The evolution of photosynthesis freed life from dependence on abiotic organic compounds. Oxygenic photosynthesis, which produces oxygen as a byproduct, was particularly revolutionary. It changed Earth's atmosphere and oceans, creating new opportunities and challenges for life. The Great Oxidation Event, around 2.5 billion years ago, was a catastrophe for many organisms adapted to anaerobic conditions, but opened up new possibilities for oxygen-using organisms. The evolution of cellular respiration, which uses oxygen to efficiently extract energy from food, enabled much more energetic and complex life forms. Multicellular organisms with high energy demands became possible only after atmospheric oxygen levels rose. So the history of life is intimately tied to the history of energy flows on Earth. Communication between cells is fundamental to modern life, even in simple organisms. Bacteria can sense chemical signals from other bacteria and respond appropriately, coordinating behavior across populations. This quorum sensing allows bacteria to time certain activities for when their population is large enough to be effective. Such communication might have ancient origins. Even protocells could potentially have leaked chemicals that affected neighbors, creating primitive forms of interaction. Selection could have favored cells that responded appropriately to these signals, gradually elaborating communication systems. The ability to cooperate and coordinate across cells would have provided advantages, driving evolution of signaling mechanisms. In multicellular organisms, communication between cells becomes essential. Development of a complex organism from a single cell requires elaborate signaling to coordinate differentiation and growth. The nervous system is ultimately an elaborate intercellular communication network. Human thought emerges from communication among billions of neurons. So the social nature of life, the interaction and communication between biological units, has deep roots extending back perhaps to the earliest cells. The mineral composition of early Earth's oceans and atmosphere would have profoundly affected prebiotic chemistry. Different minerals catalyze different reactions. The presence or absence of certain elements would determine which molecules could form easily. Understanding early Earth's geochemistry is therefore crucial for understanding how life began. Some minerals form layered or porous structures that could have provided surfaces and compartments for concentrating organic molecules. Clay minerals have been proposed as possible templates for early life. Their layered structure might have helped organize molecules and catalyze polymerization reactions. Some clays can even preferentially bind certain organic molecules, potentially selecting for biochemically relevant compounds. Iron-sulfur minerals found at hydrothermal vents can catalyze organic reactions relevant to metabolism. Modern proteins called iron-sulfur clusters are used in many metabolic enzymes, possibly reflecting the ancient role of these minerals. The transition from mineral catalysts to protein enzymes was probably gradual, with early life forms using hybrid systems before eventually replacing mineral catalysts entirely with biological ones. Phosphate availability on early Earth may have been a limiting factor for life. Phosphorus is essential for nucleic acids, ATP, and cell membranes. However, phosphate is not very soluble in seawater and may have been scarce. Some scientists propose that early life used different molecules without phosphorus, later switching to modern phosphate-based biochemistry when environmental phosphate levels increased. Others suggest that life originated in environments like volcanic ponds, where phosphate was more readily available. This question remains unresolved, but highlights how environmental chemistry constrained possible paths to life. Temperature is another crucial variable. Many origin of life theories focus on hot environments like hydrothermal vents, where thermal energy could drive reactions. but heat also breaks down complex organic molecules. Other theories emphasize cooler environments where molecules are more stable. Perhaps life required both: hot regions for energy and synthesis, cool regions for preservation, with cycling between the two allowing accumulation of complex molecules. Seasonal or tidal cycles could have provided such variations. The pH level, the acidity or alkalinity of early oceans, also mattered. Different pH levels favored different chemical reactions. Modern life carefully controls internal pH, but early protocells would have been at the mercy of environmental pH. Hydrothermal vents create strong pH gradients where acidic vent fluids meet alkaline ocean water. These gradients could have provided energy and driven synthesis of organic molecules. Some origin of life theories build on these natural proton gradients as an energy source for the earliest metabolic reactions. The diversity of environments on early Earth means that many different chemistries were occurring simultaneously in different locations. Life might have originated in one particular type of environment and later adapted to others. Or multiple independent origins might have occurred, with competition eventually leaving only one survivor. The winner, the lineage leading to all modern life, may have succeeded because it originated in an environment that provided particular advantages, or simply through luck. We may never know exactly where and how life began, but by studying many possible environments and mechanisms, we can understand the range of possibilities and constrain our theories. Each experiment and observation narrows the window of plausible scenarios. Eventually, we may achieve a comprehensive understanding, even if some details remain forever obscure. The role of chance in life's origin deserves more consideration. At every step, from the formation of organic molecules to the first self-replicating system to the last universal common ancestor, chance events shape the outcome. A particular molecule happened to form in a particular location. A random mutation improved some function. A fortunate combination of circumstances allowed some protocell to thrive. These chance events, accumulated over millions of years, determined the specific path life took. Yet underlying the chance is necessity. The laws of chemistry and physics constrain what can happen. Some molecules are much more likely to form than others. Some structures are much more stable. Some functions are much more useful. Natural selection is a deterministic process acting on random variation. So life's origin involved both chance and necessity, randomness and inevitability, interacting in complex ways. This interplay continues in evolution today. Mutation and environmental change introduce randomness. Selection and physical law impose order. The result is the amazing diversity and complexity of life, neither completely predictable nor completely random, but emerging from the interaction of both elements. As we continue this peaceful exploration of life's beginning, reflect on how the universe is capable of producing life. The same atoms that form stars and planets can, under the right conditions, organize into living systems. This is not magic or miracle, but neither is it mundane. It's a natural process, but one of stunning profundity. Matter can become complex enough to reproduce itself, to evolve, to eventually think and wonder about its own origins. The path from simple atoms to human consciousness is long and winding, but it's a continuous path, each step following from previous ones through natural processes. This continuity is reassuring in some ways. We're not separate from nature but deeply embedded in it. The same principles that govern the cosmos govern us. Understanding these principles helps us understand ourselves and our place in the universe. It connects us to everything, from the smallest bacterium to the largest galaxy. We're all part of one grand story, the story of matter organizing itself into ever more complex and interesting forms. The search for life elsewhere in the universe has intensified in recent decades. We've discovered that planets are abundant, that organic molecules exist throughout space, that liquid water may be common. These discoveries increase the likelihood that life exists beyond Earth. If we find it, comparing Earth life with alien life would revolutionize our understanding of life's possibilities and requirements. If alien life uses the same biochemical mechanisms as Earth life, it might suggest that there are very few ways to build living systems, or it might suggest panspermia, spreading of life between worlds. If alien life uses completely different mechanisms, it would prove that our biochemistry isn't the only solution and would raise fascinating questions about alternative chemistries. Either discovery would be profound. For now, we have only one example of life, Earth life. But we're learning to read that example more deeply, extracting information about ancient history from modern organisms. We're recreating possible prebiotic conditions in laboratories and observing what emerges. We're exploring extreme environments on Earth and finding life in places once thought uninhabitable. Each discovery expands our conception of life's possibilities and informs our theories about its origin. The deep interconnection between all life on Earth is worth emphasizing. Every organism shares core biochemistry inherited from our common ancestor. When you eat food, your cells break it down using metabolic pathways that evolved billions of years ago. When you think, your neurons communicate using mechanisms refined over hundreds of millions of years. When you reproduce, you pass on DNA using the same genetic code used by every other living thing. These connections are not abstract. They're physical and functional. Your body is a living fossil, carrying within it structures and processes that date back to life's early history. Your mitochondria, descended from ancient bacteria, still retain their own DNA separate from your nuclear genome. The very atoms in your body have been recycled through countless organisms over billions of years. You're literally made of the same matter that formed ancient microbes, dinosaurs, and primordial oceans. This material continuity links all life across time. The atoms in your hand were once in other living things, and before that in Earth's ancient seas, and before that in dying stars. We're all temporary assemblies of atoms that will eventually disperse and reassemble into other forms. Life is a process, a pattern of organization, rather than a static thing. The molecules in your body are constantly being replaced, yet you remain you. What persists is the pattern, the information, the organization. This is true of all life. Individual molecules come and go, but the pattern persists and evolves. This perspective helps us think about life's origin. The first life wasn't a particular molecule, but a particular pattern of organization that could maintain and copy itself. Once such a pattern emerged, evolution could refine it, but the essential feature was self-maintaining organization. Modern life has incredibly sophisticated versions of this organization, but the basic principle dates back to the beginning. The question of whether life is inevitable or improbable remains open. If life arises readily wherever conditions permit, the universe may be full of it. If life requires extremely unlikely coincidences, we may be alone or nearly alone. This question has implications not just for astrobiology, but for our understanding of life's nature and significance. Some scientists argue that life is a natural consequence of thermodynamics. Living systems are very good at increasing entropy in their environment, while maintaining their own internal organization. They're dissipative structures that exist far from equilibrium, maintained by flows of energy and matter. Similar structures appear in non-biological contexts, like convection cells and tornadoes. Perhaps life is just a particularly sophisticated example of a general phenomenon where energy flows through matter and creates organized structures. If this view is correct, life may be common wherever energy gradients exist. Other scientists emphasize the many specific requirements for life and the apparent fine-tuning of physical constants necessary for chemistry to be rich enough to support life. They argue that life may be rare and precious, an unlikely outcome even given billions of years and countless planets. This debate cannot be fully resolved until we find other examples of life or definitively fail to find them despite extensive searching. For now, both possibilities remain open. What we can say is that life arose at least once, here on Earth, from non-living matter through natural processes. This fact alone is remarkable and worthy of deep contemplation. As you rest and absorb these ideas, consider the privilege of being alive and conscious. You're experiencing the universe from the inside, as it were. Your thoughts and feelings are the universe thinking and feeling through the particular pattern of organization that is you. This isn't mysticism, but rather a scientific perspective on consciousness and existence. You're a temporary eddy in the flow of matter and energy through the cosmos, granted a brief moment of self-awareness before dispersing back into the universe. This perspective can be humbling and empowering. Humbling because it reminds us we're not separate from or above nature but fully part of it. Empowering because it reveals we're capable of understanding our own origins and nature through science and reason. We can comprehend the processes that made us, trace our lineage back billions of years, and see our connection to everything. This understanding is one of humanity's greatest achievements. The practical applications of origin of life research extend beyond satisfying curiosity. Understanding how life works at its most fundamental level informs medicine, biotechnology, and agriculture. Synthetic biology uses principles learned from studying life's origins to design new biological systems. Creating artificial cells could lead to novel ways of producing medicines, materials, or energy. Understanding the minimum requirements for life helps us design life support systems for space exploration. These practical benefits, while significant, are almost beside the point. The primary motivation for studying life's origin is wonder and curiosity about ourselves and our place in nature. We want to know how we came to be because we're creatures capable of asking such questions. This capacity for wonder and inquiry is itself a product of evolution, an outcome of the same processes we're studying. Our brains evolved to understand the world, and now we're using them to understand their own origins. This self-referential quality of consciousness studying its own emergence is philosophically fascinating. We're part of the universe that has become complex enough to contemplate itself, to ask questions about its own history and nature. Whether this has happened elsewhere, we don't know. But it's happened here, and we're living through this moment of cosmic self-awareness. Future generations will know more about life's origin than we do. Research continues, technology improves, and our understanding deepens. Perhaps someday we'll fully understand each step from simple molecules to living cells. Perhaps we'll even recreate the process in the laboratory, demonstrating that life can indeed emerge from chemistry. Such knowledge won't diminish life's wonder but will enhance it. Understanding how something works deepens appreciation of its elegance and complexity. Knowing how eyes evolved doesn't make vision less amazing. Knowing how brains work doesn't make thought less remarkable. Similarly, understanding how life originated will help us appreciate the beautiful necessity of natural processes, while marveling at the specific path history happened to take. For now, we live with uncertainty and mystery, which has its own beauty. The questions about life's origin remain among the deepest and most fascinating in science. Each generation of scientists chips away at these questions, and each generation of people can contemplate the answers and wonder at existence. As you drift towards sleep, let your mind rest with these thoughts about life's deep history and the processes that made you possible. Feel connection with all living things, knowing we share common ancestry. Feel connection with Earth, knowing we're made from its materials and shaped by its history. Feel connection with the universe, knowing we're made of stardust and governed by the same laws that govern galaxies. These connections are real, not metaphorical. You are quite literally part of an ancient and ongoing story, the story of matter becoming alive and aware. Rest well in this knowledge, comfortable in your place in nature's grand tapestry, one thread among countless others, unique yet connected to all. Sleep peacefully and dream of deep time, of ancient oceans, of the first cells swimming in primordial seas, of evolution's long dance leading from there to here, to you. To this moment of consciousness and wonder. Good night and sweet dreams of life's magnificent origins and endless possibilities. The story continues, as it has for billions of years, and you're part of it, here, now, always. Rest now, rest deeply, and let these thoughts settle gently like sediment on an ancient sea floor, layer by layer, building understanding slowly, naturally, peacefully. Let us explore even deeper into the mysteries of life's beginning, examining more closely the molecular mechanisms and evolutionary innovations that made life possible. The structure of DNA itself tells us much about life's history and origins. DNA is a double helix, two strands wound around each other with complementary base pairing holding them together. This structure, discovered by Watson and Crick in 1953, immediately suggested how DNA could replicate. Each strand serves as a template for making a new complementary strand. But this elegant structure didn't arise fully formed. It evolved from simpler precursors. RNA, chemically similar to DNA but with subtle differences, is more versatile and may have preceded DNA. RNA uses the sugar ribose while DNA uses deoxyribose, lacking one oxygen atom. This small change makes DNA more chemically stable, better suited for long-term information storage. Early life may have used RNA for genetics, before evolving DNA for its superior stability. The transition from RNA to DNA was a major evolutionary innovation that improved information preservation across generations. The four bases used in nucleic acids, adenine, guanine, cytosine, and thymine in DNA or uracil in RNA, are not arbitrary choices. These particular molecules have chemical properties that make them ideal for genetic information storage. They can form strong but reversible bonds with each other through hydrogen bonding. They're relatively stable but can be synthesized from simple precursors. They absorb ultraviolet light, which might have helped protect early genetic molecules from UV damage. The specific pairing rules, adenine with thymine or uracil and guanine with cytosine, arise from the shapes and bonding patterns of these molecules. This complementary base pairing is crucial for accurate replication. A template strand specifies exactly which nucleotides should be added to the new strand. Errors do occur, creating mutations that fuel evolution, but most of the time the pairing rules ensure faithful copying of genetic information. This balance between fidelity and variation is essential for evolution. Perfect copying would prevent adaptation. Too many errors would destroy useful information. The actual error rate in modern DNA replication, about one mistake per billion nucleotides, reflects billions of years of optimization by natural selection. Early replicators likely had much higher error rates, making evolution faster but also more difficult. The evolution of error correction mechanisms was probably crucial for allowing genomes to grow larger and accumulate more complex information. Modern cells have elaborate DNA repair systems that fix mistakes and damage. These systems themselves are encoded in DNA, creating a self-maintaining informational system. This chicken and egg situation suggests that early life forms had simpler repair mechanisms that gradually became more sophisticated. Each improvement in information preservation allowed more complex adaptations to evolve, driving life toward greater complexity. The relationship between genotype, the genetic information, and phenotype, the physical expression of that information, is fundamental to life. Natural selection acts on phenotypes, but evolution occurs through changes in genotypes. This separation allows evolution to work because useful variations can be preserved in DNA and passed to offspring. The origin of this genotype-phenotype distinction was crucial for evolution's power. In modern life, RNA serves as intermediary between DNA and proteins, transcribing genetic information and translating it into protein sequences. This central dogma of molecular biology, information flowing from DNA to RNA to protein, organizes all cellular life. But in the RNA world, before DNA and proteins, this distinction may have been blurred. RNA molecules served as both genotype and phenotype, both information storage and functional molecules. The later evolution of specialized molecules for each role, DNA for information and proteins for function, improved efficiency and capabilities, but built on the foundation established in the RNA world. The genetic code's redundancy, with multiple codons specifying the same amino acid, provides a buffer against mutations. A change in the third position of a codon often doesn't change which amino acid is incorporated. This wobble-base pairing reduces the impact of mutations, making genetic information more robust. The code structure seems optimized for error tolerance, suggesting it was refined by natural selection over long periods. Alternative genetic codes are possible and have been found in some organisms, but they're rare. Once a code is established in a population, changing it is nearly impossible without breaking everything that depends on it. This lock-in effect means the genetic code we inherited from the last universal common ancestor has persisted for billions of years. It's a frozen accident, one of history's contingent details that became permanent. The size of early genomes was probably tiny compared to modern organisms. The smallest known independent genomes today are those of certain bacteria, containing about 500 genes. But the first replicators may have had just one or a few genes, if we can even call them genes. A single ribozyme capable of copying itself would constitute a minimal replicator. From such humble beginnings, genomes grew through duplication, mutation, and acquisition of new genes. Each functional addition that improved survival or reproduction was preserved by selection. Over billions of years, genomes expanded from perhaps a few dozen nucleotides to millions or billions. The human genome contains about 3 billion base pairs encoding roughly 20,000 genes. This vast expansion happened gradually through evolutionary processes. Not all genome growth represents new information. Much DNA is non-coding, meaning it doesn't specify proteins. Some non-coding DNA has regulatory functions, controlling when and where genes are expressed. Other portions are evolutionary fossils, remnants of ancient genes or viruses. Still others may have no function at all, just along for the ride because they don't harm their host enough to be removed by selection. The nature and amount of non-coding DNA has been debated vigorously. Some scientists argue most DNA is functional even if we don't understand its purpose yet. Others contend that much of it is junk, accumulated through neutral evolutionary processes. The truth likely lies between these extremes. Some non-coding DNA is clearly functional. Some is clearly junk. Much remains uncharacterized. This complexity reflects billions of years of evolution building on previous structures without discarding them. Life doesn't start fresh with each generation, but inherits the accumulated baggage of evolutionary history. This makes biology messy and complex, but it also provides clues about the past. By studying what's been preserved, we learn about ancient life and the processes that shaped modern organisms. Viruses complicate our understanding of life's origin and nature. Are viruses alive? They can reproduce, but only inside host cells. They evolve, showing adaptation to changing environments, but they lack metabolism and can't reproduce independently. This makes their classification ambiguous. For understanding life's origins, viruses are interesting because they represent minimal replicators, genetic information in a protein coat. Some viruses use RNA as genetic material, potentially resembling early replicators. Giant viruses blur the line between viruses and cells, having genes that typical viruses lack. The discovery of these complex viruses has prompted reconsideration of how viruses relate to cellular life. Did viruses evolve from cells by losing functions? Or are they descendants of ancient procellular replicators that never evolved into full cells? Different types of viruses may have different origins. The relationship between viruses and cells has been intertwined throughout life's history. Some viruses insert their genes into host genomes, becoming permanent parts of their host's DNA. About 8% of human DNA comes from ancient viral insertions. These endogenous viruses are evolutionary fossils, evidence of past infections. Occasionally, they've been co-opted for useful functions. Some genes essential for placental development in mammals came from ancient viruses. This demonstrates how evolution works with available materials, repurposing existing structures for new functions. The distinction between living and non-living becomes fuzzy at life's edges. Viruses, prions, and viroid RNAs all show some lifelike properties but lack others. This fuzziness isn't a problem for science, but rather reflects nature's continuity. The categories we impose, living versus non-living, are human constructs that may not carve nature at its joints. At life's origin, there must have been systems that were neither clearly alive nor clearly not alive. The emergence of life was gradual, not a sudden transition. Understanding this gradual emergence requires studying systems that exist in that gray zone. The role of cooperation in early evolution deserves more attention. While we often think of evolution as competitive struggle, cooperation has been equally important. The endosymbiotic origin of eukaryotes, where one cell took up residence in another, is cooperation par excellence. The genetic code itself reflects cooperation among molecules. Transfer RNAs cooperate with messenger RNA and ribosomal RNA to build proteins. Metabolic networks involve many enzymes working together in coordinated sequences. Even at the earliest stages, cooperation probably mattered. Autocatalytic sets, where molecules help catalyze each other's formation, involve molecular cooperation. Protocells that could share resources or products might have thrived better than isolated ones. The evolution of multicellularity took cooperation to new levels. Individual cells in a multicellular organism cooperate so extensively that they function as a single entity. This transition from unicellular to multicellular life happened multiple times independently in different lineages, suggesting it provides major advantages. But it also requires solving cooperation problems. Cells must suppress their individual reproduction in favor of the organism's reproduction. This requires mechanisms to prevent cheating cells that selfishly reproduce at the expense of others. Such cheating cells are essentially cancer. Multicellular organisms evolved elaborate mechanisms to suppress cancer and maintain cooperation among cells. These mechanisms include cell-cell communication, programmed cell death, and immune surveillance. The evolution of complexity often requires new solutions to cooperation problems. The speed of chemical reactions matters greatly for life. Most organic reactions are very slow at biological temperatures without catalysts. Enzymes speed up reactions by factors of millions or billions, making life's chemistry possible. How did early life get fast enough reactions without sophisticated enzymes? Mineral catalysts provided some help. Ribozymes could catalyze certain reactions. Perhaps early life just had very slow metabolism compared to modern organisms. Protocells might have grown and divided only once per year or even longer. As catalysts improved through evolution, life could speed up, allowing faster growth and reproduction. Faster reproducers would outcompete slower ones, creating selection pressure for better catalysis. Over time, metabolic rates increased from geological timescales to the rapid pace we see in modern microbes, some of which can double in population every 20 minutes under favorable conditions. The evolution of enzymes from ribozymes probably happened gradually. Some modern enzymes still use RNA cofactors, perhaps reflecting their evolutionary heritage. The ribosome, as mentioned earlier, uses RNA to catalyze peptide bond formation, even though it's surrounded by protein. This suggests ribozymes came first, and proteins were added later to improve performance. The vastly greater diversity of protein shapes compared to RNA structures explains why proteins took over most catalytic roles. The 20 amino acids used in proteins have diverse chemical properties, allowing proteins to be shaped into countless forms specialized for different functions. RNA, built from only 4 nucleotides, has more limited structural possibilities. Still, the discovery of ribozymes showed that RNA is more capable than once thought, supporting the RNA world hypothesis. The origin of the 20 amino acids used in proteins is another puzzle. Why these 20? Other amino acids exist and are sometimes used for specialized purposes, but they're not standard. The standard 20 may have been selected because they're easily synthesized, chemically diverse, and unlikely to cause harmful side reactions. Or they may be partially arbitrary, the set that happened to be incorporated early and then became frozen in place. Laboratory experiments creating artificial proteins with non-standard amino acids show that alternative biochemistries are possible. But once life committed to a particular set of building blocks, changing became nearly impossible. All the cellular machinery for making and using proteins evolved to work with the standard 20 amino acids. Switching to a different set would require redesigning everything simultaneously. This explains why biochemistry is so conservative, preserving ancient choices for billions of years. The handedness of molecules, discussed earlier as chirality, also applies to sugars and amino acids in specific ways. DNA and RNA use right-handed sugars exclusively. Proteins use left-handed amino acids exclusively. These choices seem arbitrary. Mirror-image life using opposite chiralities would work equally well. But within a living system, consistent chirality is essential. Mixed chirality would prevent the regular helical structures of nucleic acids and proteins. So life had to choose one or the other, and the choice we see is what happened to be selected billions of years ago. If we found life that used opposite chirality, it would strongly suggest an independent origin. As it's unlikely that two life forms with such fundamental biochemical differences could share a recent common ancestor. Lipids and membranes also show interesting patterns. Cell membranes in bacteria and eukaryotes use lipids based on glycerol with fatty acids attached. Archaea use different lipids with branched chains and ether linkages instead of ester linkages. This suggests bacteria and archaea diverged early, before their membrane composition was finalized. Or perhaps the last universal common ancestor had relatively simple membranes, and different lineages independently evolved more sophisticated ones. Membranes do more than just enclose cells. They're platforms for many cellular processes. Proteins embedded in membranes perform functions like energy generation, nutrient transport, and sensing environmental signals. The evolution of membrane proteins was crucial for increasing cellular capabilities. Early protocells with pure lipid membranes would have been limited in what they could do. Adding membrane proteins opened up new possibilities, but it required coevolution of membranes and proteins to work together. This is another example of how different cellular systems had to evolve in concert. The proton gradients across membranes that power ATP synthesis, the energy currency of cells, are universal in cellular life. This suggests proton-powered energy generation dates back to very early life, perhaps even before the last universal common ancestor. Some theories propose that natural proton gradients at hydrothermal vents were co-opted by early cells, which later evolved their own mechanisms to generate gradients. Modern cells use elaborate protein complexes to pump protons across membranes and then harvest the energy as protons flow back down their gradient. This chemiosmotic mechanism is fundamental to biology. Its universality indicates ancient origins and fundamental importance. The evolution of sensory capabilities allowed organisms to respond to their environment more effectively. Even simple bacteria can sense chemicals and move toward or away from them. This chemotaxis requires sensing molecules, signal transduction pathways, and motility mechanisms. The evolution of these systems enhanced survival by helping organisms find food and avoid dangers. More complex organisms evolved increasingly sophisticated sensory abilities: vision, hearing, touch, taste, smell. These senses provide information about the environment that can guide adaptive behavior. The nervous system processes this information and coordinates responses. The brain, in complex animals, creates internal models of the world that can be used for prediction and planning. This progression from simple chemical sensing in bacteria to human consciousness shows how evolution can build increasing complexity on ancient foundations. Each new capability adds to what existed before. The molecular mechanisms underlying sensation are related across organisms. Light-sensing proteins in human eyes are related to light-sensing proteins in bacteria, showing common ancestry. The calcium ion signaling used in neurons is related to calcium signaling in single-celled organisms. Evolution works by modifying existing structures and mechanisms, not by creating entirely new ones from scratch. This principle, called descent with modification, was recognized by Darwin and remains central to evolutionary biology. In the context of life's origins, it means early cells didn't need to evolve everything from scratch. They inherited some structures and processes from prebiotic chemistry. They built on the chemical foundations that preceded them. This continuity from chemistry to biology eases the path from non-life to life. Understanding the minimal requirements for life helps us recognize this continuity. What's absolutely necessary for something to be considered alive? Different scientists emphasize different features: metabolism, reproduction, evolution, compartmentalization, homeostasis, response to stimuli. Modern Earth life shows all these features. But at life's beginning, some features may have been absent or rudimentary. Drawing a sharp line between living and non-living may be impossible and unnecessary. What matters is understanding the continuum and how systems transitioned along it. The study of extreme environments on Earth has revealed organisms living in conditions once thought incompatible with life. Thermophiles thrive in boiling water. Psychrophiles grow in perpetual ice. Acidophils live in extremely acidic environments. Halophiles require high salt concentrations. These extremophiles expand our conception of life's possibilities and suggest that life's origin might have occurred in what we would consider extreme conditions. Perhaps the first cells were thermophiles living near hot springs or hydrothermal vents. Modern extremophiles may preserve some characteristics of ancient life, adapted to environments that resemble early Earth more than modern temperate surface conditions. By studying these organisms, we gain insights into what early life might have been like and what mechanisms it used to cope with challenging environments. The deep biosphere, life existing kilometers below Earth's surface, is another frontier. Microbes in rock pores and cracks survive on minimal energy from geochemical reactions. These ecosystems are isolated from the surface, receiving no sunlight and no input from surface biology. They've existed for millions of years, evolving independently in the dark. Some scientists speculate that life might have originated in the subsurface, protected from surface impacts and radiation. The discovery of the deep biosphere shows that habitable environments exist even without surface conditions we typically associate with life. This has implications for potential life on other worlds. Mars likely has underground liquid water. Europa and Enceladus have subsurface oceans. These environments could potentially host life even though their surfaces are inhospitable. As we relax and ponder these mysteries, consider how much we've learned about life's origins in just a few decades of intensive research. We've gone from vague speculation to testable hypotheses, from crude experiments to sophisticated simulations, from ignorance to genuine understanding in many areas. Yet much remains unknown, and the deepest questions remain unanswered. This combination of knowledge and mystery is intellectually exciting. We know enough to frame precise questions and design informative experiments. We don't know enough to be certain of answers. This is the sweet spot for scientific inquiry, where progress is possible and discoveries await. Future breakthroughs might come from unexpected directions. Perhaps computational simulations will reveal previously unknown chemical pathways to life. Perhaps studies of exoplanets will show how common habitable environments are. Perhaps laboratory experiments will achieve synthesis of self-replicating systems, demonstrating a pathway from chemistry to life. Perhaps exploration of Mars or icy moons will find evidence of independent origins of life, showing it's not unique to Earth. Each of these possibilities could revolutionize our understanding. Each is being actively pursued by researchers worldwide. The global scientific community is gradually piecing together the story of how life began, contribution by contribution, discovery by discovery. One important aspect we haven't fully explored is the role of information in life. Living systems process information in profound ways. DNA stores information about how to build an organism. RNA transfers this information. Proteins use it to perform functions. Organisms sense their environment, processing information about external conditions. Brains integrate sensory information to guide behavior. Information processing is arguably as fundamental to life as energy and matter. The origin of biological information is thus a key question. How did meaningful information arise in molecular systems? Information in this context means sequences that specify function. A gene is informative because its sequence determines a protein's shape and function. Random sequences carry no information. The origin of information requires some process that generates functional sequences. Natural selection is such a process. Random variation creates different sequences. Selection preserves those that work. Over time, functional sequences accumulate. This requires reproduction with variation, so selection can act. But it also creates a chicken and egg problem. Information seems necessary for reproduction, but reproduction seems necessary for generating information. The RNA world hypothesis addresses this by proposing that RNA molecules could both carry information in their sequences and perform functions through their three-dimensional shapes. A lucky RNA sequence that could catalyze its own replication would embody both information and function. Such a molecule could reproduce with variation, allowing evolution to refine and improve it. The information content would increase as selection favored more efficient replicators. Eventually, enough information would accumulate to specify complex functions like metabolism and membrane synthesis. This scenario makes the origin of information a gradual process, starting with minimal information in a simple replicator and building up through evolution. Each beneficial mutation adds a bit more information. Over geological time, genomes grow from perhaps dozens of nucleotides to millions or billions. The information in a human genome, if written out as text, would fill thousands of books. All this information arose through evolutionary processes acting on variation and preserving what worked. No intelligence or foresight was required, just the blind action of natural selection over vast time scales. This is one of evolution's most remarkable features, its ability to generate complex information and functionality through entirely natural, material processes. The origin of life is thus inseparable from the origin of biological information. Understanding how meaningful sequences could arise from chemistry is crucial to understanding how life began. Shannon information theory, developed for communications engineering, has been applied to biology with interesting results. This theory quantifies information content mathematically. Genes can be analyzed for their information content. Genomes can be compared to measure how much information differs between species. These analyses show that information has indeed increased over evolutionary time. Early life had less genomic information than modern life. The capacity to store and process information has grown, enabling more complex organisms. This growth of information is driven by the interplay of mutation, which generates new information, and selection, which preserves useful information and discards harmful changes. The net result, averaged over long time spans, is accumulation of useful information. This doesn't happen steadily but episodically, with bursts of innovation followed by long periods of stasis. Mass extinctions can erase accumulated information, setting back evolutionary progress, but life always recovers and continues to diversify. The overall trend over billions of years has been toward greater complexity and information content, though with many reverses and exceptions. Not all organisms become more complex. Many successful lineages simplify, losing genes and functions they no longer need. Parasites often lose metabolic genes, relying on their hosts instead. Organisms in stable environments may not need complex adaptations. Simplification is as much part of evolution as increasing complexity. But the maximum complexity achieved by any organisms has increased over time. Early life was all simple prokaryotes. Later, eukaryotes evolved with greater internal complexity. Later still, multicellular organisms appeared with coordinated cells forming tissues and organs. The most recent evolution saw nervous systems and brains, with information processing capabilities far beyond single cells. This increase in maximum complexity shows evolution can generate genuinely new capabilities, not just variations on themes. The ethical implications of origin of life research are worth considering. Creating artificial life in the laboratory could have profound consequences. If we succeed in making self-replicating cells from scratch, we demonstrate that life is a natural phenomenon arising from matter and energy without any special vital force. This materialist view of life is now mainstream in science, but successfully creating life would provide definitive proof. Such proof might affect how people think about life's meaning and value. Some worry it could lead to devaluing natural life if we can make it artificially. Others argue it would increase our appreciation of life's mechanisms and complexity. These are philosophical and ethical questions without simple answers. Beyond philosophy, artificial life could have practical risks and benefits. Synthetic organisms designed for specific purposes could produce medicines, clean up pollution, or generate energy. It also grounds us in reality, helping us see our place in nature accurately rather than through comforting illusions. We're part of nature, subject to the same natural laws as everything else. Yet we're also special, at least from our own perspective, the only known species capable of understanding its own origins through science and reason. This combination of humility and pride, seeing ourselves as both products of nature and unique within it, is the appropriate stance given what we know about life's history. We should appreciate our connection to all life and to the cosmos while also valuing what makes us distinctive. Good night and sleep peacefully in the knowledge that you're part of the grand story of life in the cosmos, a story that began billions of years ago and continues in you now, this moment. Forever connected to everything that was and all that will be.